So what we did was some RNAseq, all right? So we're looking at, we did some viral transduction of some C2C12 myotubes. We used the HERG potassium channel, which is what I've been working with all along. And we have found that it induced the expression of a lot of different genes, some of them related to atrophy, as I've been describing. So just briefly, the potassium channel ERG, it's a tetramer of 4 subunits. That's the typical voltage gated channel. It's got 6 transmembrane domains, with the 4th being the sensor, and there being a pore in the center exclusively for potassium. There are 2 splice variants, the 1A, the 1B. The 1A and the 1B, when they are expressed together, make a heteromultimer that's found in the cardiac tissue. What it does is it contributes to the late phase of repolarization, and it helps to change the shape of the waveform of the heart. All right, so what we found out was that it's in skeletal muscle. It had not been reported until we started working with atrophic skeletal muscle. We found it by IHC, and we see here that it was in the suspended and after 4 days, and in the animals that were suspended for 7 days. We don't see it in the non-suspended animals using the DAB. We did some Western blotting, and again, we find that in the suspended, not in the controls. What we have found since is that we can find it in normal tissue, but we have to use some fairly sensitive ways of doing it. We use a tyramide amplification with immunohistochemistry, and then we highly concentrate the sample with immunoblotting. Okay, we do know that ERG induces atrophy. We've shown that before. When we put ERG into the muscle by electrotransfer, what we find is that the cross-sectional area decreases. We got a significant 15.3% decrease in some of the animals with which we were working. When we put a dominant negative form, a dysfunctional form of the channel into the muscle by electrotransfer and suspended the animals, what we found is that the defective expression of the defective channel would rescue the muscle size and take it back to 96% of control. We could do the same thing with suspended animals who were given a drug that blocks ERG. And what we found is that in the weight-bearing animals who got this same drug, there was actually a slight, non-statistically significant increase in the cross-sectional area of the muscle, suggesting that it is indeed in normal muscle. It's just very, very low levels, why it was never detected before. What does it do? What role does it play? What we have reported is that it increases proteolysis. We've shown that it increases the ubiquitin proteasome pathway activity, and it also increases intracellular calcium levels in C2C12 cells. As a consequence of that, we have shown that it also increases calpain activity. When we electrotransfer the channel into the muscle, what we find is that in vivo, we also get a decrease in calpain activity. So ERG is affecting proteolysis both by UPP and by modulating calcium levels. We don't know how at this point. We do know that it does cause an increase in some of the E3 ligases that are part of the UPP pathway, specifically MERF. All right. Now, one of the interesting things about ERG, the ERG1A, which is the one we find in muscle, is that it's also detected in brain and connected with epilepsy. It's also found in smooth muscle and pancreas tissue, where it's connected with mainly malignancy. It's also reported in certain cancer cells. So what exactly overall is it doing? It looks like it's affecting growth in addition to causing some degradation. So it may just be a matter of concentration of the amount of the channel in the membrane. So we decided to do some RNAseq. So we looked at C2C12 myotubes. We used a control adenovirus to express in the GFP marker in some of the cells. Then we used a HERG encoded adenovirus to express our channel in another set. After 48 hours, we extracted the RNA and sent that off for RNAseq to a genomics group in Purdue University, where my colleague Greg Hockerman works. They reverse transcribed the RNA into a cDNA library, to which trimming was done to remove bases of lower quality, those with a Phred score below 30. The resulting reads were mapped against a reference genome, a mouse genome, using a STAR program. And these were used as input for HT-seq to obtain read counts and a count matrix. This matrix is used for downstream differential gene expression analysis. That is, we determined using R which genes were exposed in the control versus the treated. We did pairwise analysis and revealed that ERG1A expression in these myotubes significantly affected about 400 different transcripts. So we've been going through this, trying to look at some that are more interesting. This is an example of the data. You've probably seen this before. There's the gene ID, there's the fold change, the p-value, the description over there. It's a lot of data we've been swimming around in. Some of the most interesting that I think we found is the fact that ERG seems to affect retinol signaling, retinoic acid. Retinoic acid has been found to play an important role in maintaining insulin sensitivity and promoting glucose uptake and use in skeletal muscle. We see here retinol then combines with its receptor. It is converted in a non-genomic pathway to retinol by SRP35, which is converted to retinoic acid by an aldehyde. This then goes and affects the genomic pathway. It leads to transcription of IGF-1, HBEGF, and decarin genes. All of these are work together to modulate the activity of the glucose 4 receptor, the GLUT4 receptor. What we found is there was a decrease, an overall decrease in a number of these genes. These shown here by the star. These that are part of the non-genomic and part of the genomic pathway. So we thought that what we're seeing here is that expression in the myotubes is linked to some derangements of skeletal muscle retinoic acid production and signaling, which makes sense because one of the things that would happen during atrophy is there would be a decrease in anabolism. So in conjunction with the ERG1A mediated activation of proteolysis, we would see there then a loss of retinoic acid and autocrine factor production, which could suppress muscle anabolism. Reactive oxygen species are also known to modulate degradation. For example, they will increase UPP activity shown here. Reactive oxygen species brought about by the P38 pathway, leading to activation of the CHOP promoter, which will then lead to transcription of MERF. It can also, reactive oxygen species can increase calpain activity by activation of TRP channels, so that it does have an effect on proteolysis. It increases proteolysis. What we find is there's an increase in response to ERG in superoxide dismutase, amine oxidase, aldoketo reductase, all of these different aldehyde dehydrogenase, all of these enzymes that will produce reactive oxygen species, which leads us to think that there is probably some modulation of reactive oxygen species by ERG, leading to this activity, increased activity of the UPP pathway and the increased calcium levels. All right, we also saw quite a bit of interferon gamma related genes that were affected. Interferon gamma is known to influence skeletal muscle homeostasis and repair, but whether the effects are positive or negative appears to be concentration and cofactor dependent. That is, concentration of the interferon itself. Exogenous interferon has been shown to improve the healing of skeletal muscle and limit fibrosis. Endogenous IFN is known to help there be efficient muscle regeneration. However, higher concentrations of it are known to inhibit myoblast and fusion of myoblast into myotubes. So is it possible that ERG is affecting these interferon-related genes? Well, it seems to be that it's increasing them, which makes sense. If the higher levels then inhibit muscle growth, then if ERG is causing there to be an increase in these related peptides, these related proteins, then it may be another mechanism by which ERG is affecting skeletal muscle degradation. There are other genes associated with modulation of the skeletal muscle structure. MERF is one of the ones we talked about with the UPP, but also the ubiquitin-specific peptidase 18, ubiquitin-like modifier activating enzyme, folliculin interacting protein, follistatin, which is a myostatin antagonist. Also, genes associated with inflammation, apoptosis, and cell growth are affected. Most of these we see are downregulated, or very moderately increased. Mesoderm-specific transcript, integrin subunit, and there are a number of these ITGs actually that are affected. The LTBP, which is involved with pancreatic cancer, lymphocyte antigen, myocyte enhancer factor, which is a transcription enhancer. Cell division cycle 45 gene is increased in terms of its transcription. BCL2, which is suppresses apoptosis, is increased. So it appears that ERG has a really broad effect and may actually act as a coordinating type protein on the membrane. So in summary, ERG1 expression in C2C12 myotubes affects expression of genes connected with the UPP, the cytokine, interferon, retinol signaling, reactive oxygenation species, all of which are connected with muscle atrophy.
5 minutes left.
I'm just finishing up here. So anyone who is interested in obtaining any of this data, I will send you the raw data, any of the tables, the talk, whatever you need, because this was supported mainly by the Department of Defense and some of the Southern Illinois University School of Medicine, and I am obligated then to share this. We are working on a paper, and we're going to get that put together and out, hopefully, with all the craziness, hopefully by this summer. I want to thank my collaborators, Dr. Hockerman, Dr. Davey, and Yoti Yimapuran, the graduate students, Clayton and Abanoff, who helped out, undergraduates, Luke, Britton, and Ashley.